Hello everyone. This presentation is an analysis of Dr. S. Secchi's Effects in Dogs with Behavioral Disorders of a Commercial Nutraceutical Diet on Stress and Neuroendocrine Parameters. The members of the group are Sam, Chelsea, Mike, Hugh, Emma, and Ariana.
The goal of Dr. Secchi's research was to evaluate the use of nutrition in treating behavioral disorders in dogs by comparing levels of neuroendocrine hormones and stress biomarkers in dogs that were fed a nutraceutical diet compared to dogs fed a controlled diet.
Just to give you an overview of the study, one of the values being studied in this article are the neuroendocrine hormones. Serotonin, dopamine, and beta endorphins are associated with calm, content, and happy behaviors, while cortisol and noradrenaline are associated with stress. Another value that was used were stress biomarkers. Oxidative stress produces free radicals, and these free radicals can be produced during the inflammatory process in cells, drug metabolism, and when fear and anxiety-related behaviors are present. For these reasons, the evaluation of specific oxidative stress-related factors, such as derivatives of reactive oxygen metabolites, also known as DROMs, and biological antioxidant potential, BAP, are done. DROMs measure the oxidant level within the blood, and the BAP measured the amount of antioxidants within the body, such as glutathione peroxidase. Both diets fulfilled the recommendations for protein, carbohydrate, and fat regarding daily requirements. Both were kibble-form, and they both had the same amount of vitamins, trace minerals, and amino acids. The main difference is that the nutraceutical diet had an added supplement tablet to the diet. The tablet was 60 to 80% hydrolyzed protein of fish and vegetables, and then 20 to 40% were Punica granatum, Valeriana officinalis, Rosmarinus officinalis, Tilia species, Crataegus oxyacantha, all of which are from plants, and then there was also green tea extract high in L-theanine and L-tryptophan in the tablet as well. Punica granatum is used to treat anxiety and insomnia. Valeriana officinalis is used to treat mild sleep disorders. Rosmarinus officinalis, Tilia species, and Crataegus oxyacantha are all anti-anxiety and antidepressant herbs. Theanine is shown to play a role in reducing stress and decreasing heart rates in chronic anxiety patients in humans, and tryptophan has been shown to have anxiety, depression, and even aggression when not in high enough levels. The study was conducted with three important aspects. There was a group of 69 dogs that were all dogs of varying breeds, also known as mixed breeds, and they were randomly assigned to either the control group or the nutraceutical group. There was also a mix of genders, so there was not an even amount of males or females in each group. The feeding guidelines were discussed on the last slide, but to reiterate, they used the same dry kibble, but one had an added supplement. They were fed the amount according to the manufacturer's label. To find out the level of neuroendocrine hormones, blood samples were collected from the cephalic vein in all animals on the first day, which is day 0 at the start before feeding the diet, and day 45, which is when they ended the study, for both the control and treatment group.
The graphs represent the levels of serotonin and dopamine when the study started at day 0 and ended at day 45. As you can see, the level of serotonin remained the same in the animals eating the controlled diet and increased in the animals eating the nutraceutical diet. The levels of dopamine decreased in those on the controlled diet and increased in those on the nutraceutical diet. Increased levels of serotonin are known to decrease aggression, while increased levels of dopamine are known to increase the feeling of satiety, which are beneficial. This graph represents the levels of noradrenaline and cortisol when the study started at day 0 and ended at day 45. The levels of noradrenaline remained constant pre- and post-treatment in the controlled diet, but decreased with the animals that ate the nutraceutical diet. The levels of cortisol also remained constant with the controlled diet and decreased with the nutraceutical diet. Cortisol and noradrenaline are commonly associated with stress. Therefore, having a decrease in these neurotransmitters is beneficial to the animal. This graph depicts the results of the levels of beta endorphins at the start of the study on day 0 and the end of the study on day 45. The animals with the controlled diet exhibited no change in beta endorphins, while the animals with the nutraceutical diet showed a significant increase in beta endorphins. Increased levels of beta endorphins are thought to promote good feelings of reward. The graph represents the results of DROM and BAP levels in the controlled and nutraceutical diets on starting day 0 and ending day 45. The level of DROM remained consistent in the pre- and post-treatment of the dogs eating the controlled diet and decreased in dogs eating the nutraceutical diet. The level of BAP remained consistent in both dogs eating the controlled and nutraceutical diet. The decreased levels of DROMs are said to contribute to feelings of less anxiety and depression. The levels of measured biomarkers in the blood of sampled animals was taken before the start of the study and at the end of the 45 days. At the end of the study, there was not a significant statistical difference in any of the parameters measured within the control. The differences in the nutraceutical diet were previously explained. Part of the study's flaws were the low sample size of 69 dogs. An ideal study has as big as a sample size as possible. For example, about 300 minimum could be considered better. There was not an equal male to female ratio, and it was not mentioned whether the dogs were altered or intact. Both of these factors together could have effects on initial biomarker levels in the dog. This test also only used dry kibbles. Assessing nutraceuticals in wet food could eliminate another variable and add support to the research. The manufacturer's suggested feeding amount was used instead of RER, which can lead to overfeeding, therefore different amounts of nutraceuticals to each patient. Because multiple nutraceuticals were used, it's unclear which had the most positive effects or if they had a synergistic effect. The researchers did not draw blood samples when the owners changed back to regular food to assess how long the effects of nutraceuticals lasted. Also, if the owners were surveyed about their dog's behavior before and after the study, we could assess if the biomarker changes correlate to true behavioral modification. The results obtained in this study add and support previous research to demonstrate that nutrition can affect animals' behavior by regulating homeostatic equilibrium of hormones and neurotransmitters. In this study specifically, the nutraceutical diet was highly tolerated without any adverse effects. The diet was responsible for the increase of neurotransmitters such as serotonin and dopamine and the decrease of cortisol and noradrenaline. Behavioral issues are a leading cause of animal euthanasia and abandonment, but something as simple as changes in nutrition and diet can make a huge difference in modifying the behavior of an ill-mannered dog. Thank you for listening to our presentation. We hope that you all learned something valuable.